Hello, everyone. Welcome to today's webinar, Algae Talks, a joint webinar of the Biological Research and Services Laboratory and the Philippine Phycological Society Incorporated. Before we get started, let's go over a few reminders for today's event. Please be reminded that this webinar will be recorded. You will be asked for your permission for this recording. To those who are unable to attend the webinar today, or who would like to watch the lectures again, you may view the video which will be posted on the BRSL and the PPSI Facebook page. Please keep your cameras off and microphones muted to not distract the speakers and other participants. We encourage everyone to actively participate in the question and answer portion. You may send in your questions at any time during the presentation in the chat box. Your questions will be answered after all the presentations. At the end of the webinar, we will provide a link to the evaluation form that you must fill out in order to receive your certificates of attendance. Lastly, there will be a photo documentation session at the end. The photos may be posted online, so for data privacy reasons, if you do not consent to have your image captured, please keep your cameras off during the photo session and remove your profile pictures. If you also prefer to not have even your name appear in the photos, you may choose to leave the meeting at this point before the photo documentation. And to formally start with today's session, may I first call on the President of the Philippine Phycological Society Incorporated and the current Associate Professor at the National Institute of Molecular Biology and Biotechnology in UP Diliman. To give his welcome remarks, please give a round of virtual applause to Dr. Alberto D. Monatilia. Sir?
Okay. So, good morning, everybody. Can you hear me? Welcome to today's Algae Talks, which is a joint webinar by the Biological Research and Science Laboratory of the UP Diliman, and then, of course, the Philippine Phycological Society, or PPSI. And this is only a testament that despite of our struggles and difficulties in conducting research under the new normal, we are able to provide you with basic scientific knowledge in the field of phycology. And at this point, we are thankful to our young and new phycologists who will be our resource speakers for this morning, and also to all the participants in this webinar who spent time and effort to be with us this morning. So without much ado, we look forward to your participation in the tentatively scheduled webinar by the PPSI, which is tentatively scheduled in October 2021. And with that, I have to greet you mabuhay. Thank you.
Thank you so much, Sir Monitila, for the welcome message. So before we move on to the presentations, allow me to introduce to you our moderator for today. He is currently a Balik scientist of the DOST Picard, also the center head of the Verde Island Passage Center for Oceanographic Research and Aquatic Life Sciences, and an assistant professor at the College of Agriculture and Forestry at Batangas State University. Please welcome Dr. J.V. Ablanya Sacco. Sir?
Thank you. Thank you very much for the introduction. We'll proceed now to the speakers. So our first speaker is currently an assistant professor in the College of Fisheries Institute of Aquaculture in the University of the Philippines, Visayas, a former research assistant in Southeast Asian Fisheries Development Center in Iloilo. She took her bachelor degree in biology from the Central Philippine University, Iloilo, then her Master's of Science in Biology from the University of the Philippines, Visayas, and her PhD in Fishery Science at Kagoshima University, Japan. A very prolific scientist with a total of 18 research articles and counting, published in ISI and/or Scopus Index Journal, focusing on seaweed ecophysiology and cultivation. Today, she will talk about the ecophysiology of seaweeds in Western Pacific Ocean, photosynthesis and chlorophyll fluorescence. I am pleased to introduce to you a good friend of mine, Dr. Iris Goreseta Borlongan. Doc I.
Yes, good morning, everyone. Will stop others. Wait, let me share my screen. Okay, I hope you're okay. I hope you could see my screen clearly. Right. So thank you very much, Doc. Yes.
Go ahead, Doc I. Thank you.
Okay, thank you. So thank you, Doc JV, again for the kind introduction. Good morning, everyone. Thank you for taking interest in today's webinar with such great turnout. It makes me optimistic and feel encouraged to share our research despite the struggles we are currently facing. So my talk this morning is titled Ecophysiology of Seaweeds in the Western Pacific Ocean, featuring their photosynthesis and chlorophyll fluorescence. So in this title slide, you see beautiful photos of the seaweed species we have studied, some of which are distributed in the cold temperate waters of Japan, while the others occur in tropical and subtropical coastlines. Perhaps you already know the importance of seaweed community in the coastal ecosystem, but I would like to mention them again. Well, seaweeds in general serve several functions. First, they are primary producers. They carry out photosynthesis, crucial in providing the base of the marine food chain and a critical component of the cobalt carbon cycle. They form a favorable ecosystem for the survival of many marine organisms that end up eventually as human food. Like in these photos, you see sargassum sands that act as habitat and breeding grounds for fish and invertebrates living in and feeding directly on the brown alga or their associated species. Seaweeds also act as food for both small and large animals and for us humans as well. So seaweeds are indeed an important fisheries resource either harvested from natural populations or commercially cultivated for human consumption, while other species are utilized because they contain substances that can be used in food processing, cosmetics, and medicine. So over time, the demand for seaweeds grew so great that led to overexploitation of natural resource and intensive farming or domestication of marine algae worldwide. The threat of climate change on seaweed communities is also an issue that we can no longer deny. Hence, it is our responsibility to protect, improve, and help our oceans or seas to continuously flourish. And in order to do so, there is basically a need to know and understand what are the essential factors that contribute to the abundance of seaweeds. The environment of an organism includes both biotic and abiotic factors. Seaweeds mainly depend on abiotic factors, the most important being light and temperature. However, each factor has an optimal range and beyond these levels, this range may be stressful or critical for the seaweeds. Therefore, elucidating the optimum or stressful environments related to the abundance and decline of seaweeds is essential for the conservation of the coastal environment and sustainable use of the resource. So, question: how can we understand the response of seaweeds under environmental gradients? Indeed, there are many approaches, including phenology and ecological studies, culture and growth studies, but these methods may require longer periods ranging from several months to years of observation and data collection. But for photosynthesis measurements, we get to obtain immediate results as this process provides a sensitive and quick response. And this approach for the study of seaweeds responses under environmental gradients can be a powerful tool to figure out their adaptation strategies. So just a brief background of what photosynthesis is. It is the process of converting light energy from the sun into chemical energy from carbon dioxide and water. This chemical energy is stored in the form of carbohydrate molecules, which add to biomass and uh, thus result in growth. Oxygen is a byproduct of the process and is released into the atmosphere, which leads us to the next question. How do we measure photosynthesis? So photosynthesis is an easily measured process and may be estimated from the rate of oxygen production. So dissolved oxygen in the seawater medium can be measured using DO sensors. Another method for the assessment of seaweed photosynthesis is the use of chlorophyll fluorescence by amplitude modulation fluorometry or PAM fluorometry. So this has become one of the most powerful and widely used techniques for ecophysiological studies. So the status of the photosynthetic apparatus as determined by chlorophyll fluorescence is defined by several parameters, including yield, which refers to the measure of efficiency of converting photons into electron transport, which may be measured in terms of maximum quantum yield or effective quantum yield measured in continuous radiance or and also it can tell the extent to which photosystem 2 is using the energy absorbed by chlorophyll and the extent to which it is damaged by excess light or other stress factors. So today's talk will deal on our research, which generally focuses on the ecophysiological aspect of some economically important seaweeds, including kelps, carrageenophytes, and other edible red and brown algae distributed in the Western Pacific Ocean by evaluating their photosynthetic responses to temperature and irradiance based on oxygen evolution and chlorophyll fluorescence measurements. So their distinct responses were related to their adaptation to the current environment in their respective habitats, as well as to their limit of distribution in the Western Pacific. For instance, Carrageenophytes, such as Eucuma and Capophycus, are commercially cultivated in tropical countries at latitudes near 10 degrees, including Indonesia, the Philippines, Malaysia, Vietnam, Tanzania, and some countries in the Latin America and the Caribbean, which allow ideal conditions for biomass production in warm waters. So seawater temperature in the tropics is relatively stable between 28 to 30 degrees Celsius. Whereas Japan's coastline is characterized by strong temperature fluctuations between seasons and latitudes. It is also influenced by ocean currents, such as the warm Kurushu current coming from the south and the cold Oyasho current coming from the north. So the climate in Japan varies from subarctic in the north, temperate in the middle, and subtropical in the south. So the geography of Japan and the flow directions of the ocean currents along the coastal region allow the persistence of several temperate and subarctic kelps, such as the Saccharina japonica and Alaria crassifolia, as well as subtropical seaweeds like Sargassum and Cladocyphone and Myristotheca. So let me show you the results of the photosynthesis temperature experiments on these seaweeds. Based on measurements of oxygen evolution and maximum quantum yield, we obtain figures such as this. So in the case of the subarctic kelp, Saccharina angustata, temperature response on oxygenic evolution showed a single peak at around 20 degrees Celsius, which is consistent to the with the highest seawater temperature in its natural distribution in Hokkaido. Dark respiration rates, on one hand, continues to increase at higher temperatures up to a point that equals the declining net photosynthesis, and so rates become negative above this temperature. As for the temperature response on maximum quantum yield, values were steady at above zero point 0.6 at low temperatures, but started to decline above 20 degrees Celsius, and even values at 32 and 36 degrees Celsius were already zero, indicating photosynthetic to deactivation. And although 20 degrees Celsius was the estimated optimum temperature for photosynthesis, this is actually quite high as compared to the mean ambient temperature and the temperature requirements for their growth. Because based on culture experiments on related studies, optimum temperature for growth would range around 8 to 13 degrees Celsius. But this is not something unusual because we consider growth as an integration of the effect of temperature on total metabolism, including nitrogen assimilation and uptake. So perhaps we could suggest 20 degrees Celsius as the upper limit for metabolic processes, including growth of the species. In the case of the brown alga Cladocyphon omizakii, this alga is distributed in the temperate waters of Honshu, Shikoku, and Kyushu Islands, so in the middle of area or islands in Japan. Their optimum temperature for photosynthesis was around 26 degrees Celsius, which corresponds well with the summertime seawater temperatures in the distribution of these species. Their photosynthesis rates declined while respiration increases at higher temperatures, which corresponds well with the summertime seawater temperatures in the distribution of these species. You may as well have observed the discrepancy between the temperature response of oxygenic evolution and maximum quantum yield. This could be attributed to the processes being observed. Oxygen evolution represents net photosynthesis, which includes the oxygen consuming process of respiration, while maximum quantum yield reflects the gross PS2 dependent electron transport that is only part of the several processes involved in photosynthesis. Nevertheless, both maximum quantum yield and oxygen evolution measurements are significant in estimating for the lower and upper limits of temperature tolerance. Now, how about the tropical species? For example, this is Capophycus striatus. This alga was collected from Bali, Indonesia. So their optimum temperature for maximum photosynthesis is between 30 and 32 degrees Celsius. Although this temperature is within the range of seawater temperature in the cultivation site, it is likely close to the upper limit for thermal inhibition given the decline in maximum quantum yields above this temperature. As you can see in this uh, graph. This likewise is true. In fact, temperatures above 32 degrees Celsius have caused seaweed pigment concentrations to drop, leading to poor growth rates and finally to ice whitening. So they become fragile and eventually disintegrate. Hence, the seaweed production in the cultivation site would be greatly affected during the dry season when temperatures are extreme. Now, comparing the temperature optima for photosynthesis and the highest seawater temperature in the habitats of the three species, they are almost identical. But please keep in mind that we regard such temperature as the threshold level for thermal stress, as it is the result of all the metabolic processes of photosynthesis. Another interesting observation from our research that is related to temperature response of photosynthesis was the similarity in temperature optima of two heteromorphic life history stages of some seaweeds. For instance, the macroscopic sporophyte and the microscopic gametophyte stages of Cladocyphone umizakii and Saccharina japonica. While their gross photosynthesis rates are different, which could be related to the innate variability in their morphology and cellular structure, values peak along similar temperature ranges. So this further suggests the presence of both generations in the habitat throughout the year, regardless of seasonal patterns of growth and reproduction of the species. This is in contrast to another red alga known as pyropia, which have completely different temperature optima for the two stages corresponding to their seasonal occurrence. That is during summer for the sporophyte and winter during the or during winter for the gametophyte stage. Now we move on to the irradiance response on photosynthesis. But before that, let me just show the attenuation of light in the sea. So when the sun's radiation reaches the sea surface, it is attenuated by water and the intensity of light decreases exponentially with depth. So this is clearly represented by in this figure on the left after fitting the linear regression on the logarithmic scale of actual irradiances values on the right. As available light it exponentially decreases as depth increases, so does the spectral quality of light. So water molecules absorb infrared, red, and ultraviolet light first in the upper 10 to 40 meters, and then orange and yellow. Shorter wavelengths penetrate further, with blue and green light reaching the deepest depths. Hence, subtidal seaweeds need to adjust their photosynthetic machinery to maintain photosynthetic capacity under the limited light environment. Irradiance response on oxygenic evolution can be expressed in a photosynthesis versus irradiance curve, which requires a nonlinear mathematical function to account for the light saturation effect. PE parameters that can be derived from the curve include alpha, or the initial slope of the curve, Pmax, or the maximum net photosynthetic rate, dark respiration rates, and the compensation and saturation irradiances, which represent the optimum temperature for photosynthesis. There are basically two strategies for light adaptation in seaweeds related to their uh, vertical distributions. This can be graphically described by their curves. So highlight adapted species exhibit steeper slopes or higher values of alpha, higher Pmax, and saturation irradiance as compared to the low light adapted species like those seaweeds in the deeper depths of the subtidal zone or in shaded crevices or habitat under kelp canopies. They are also prone to photoinhibition, represented by a decrease in net photosynthesis rates and photosynthetic efficiency at high irradiances. This is an example of a low light adapted species. This is a red edible alga, Meristotheca populosa from Japan. Such seaweed occurs in the upper and middle subtidal zones between 3 and 30 meters deep. So its pH response showed quick saturation at around 100 micromole photons and a steeper slope or higher value of alpha. Its maximum net photosynthetic rates was likewise relatively low when we compare it with the intertidal species, and so cannot become canopy forming community in shallow waters. One more feature of Meristotheca populosa, which is a clear evidence of their low light adaptation, is their ability to optimally photosynthesize under green light. In fact, highest net photosynthetic rates were obtained from samples exposed to this wavelength. So this response is related to the spectrum distribution occurring in deep coastal waters where blue and green light prevails. It could have resulted from the absorption of green light by phycobiliproteins, specifically phycoerythrin, in phycobilisomes associated with photosystem 2, which in turn transfers the collected energy to chlorophyll centers for photosynthesis. So such premise is also supported by the increase in phycoerythrin content on samples exposed to blue and green light, as you can see in this figure. This could be attributed to the enhanced production of pigment proteins by blue and green light and to improve energy absorption to make up for the absence of other light qualities that are usually absorbed by chlorophyll. And as Japanese consumers prefer a reddish color product for these species, cultivation or post-harvest measures may be modified to achieve this target. We can also find these two light response strategies in the life history stages of seaweeds. For instance, the distinct photosynthesis irradiance responses of the two generations with higher net photosynthetic rates, alpha, and saturation irradiances on the macroscopic stage as compared to the microscopic stage. The difference in photosynthetic responses between the two life stages is related to the differences in their morphology and perhaps also its irradiance exposures within the subtidal zone. Because mature sporophytes have fully developed thalli and long blades that get higher irradiance exposures than the early life history stages that usually settle in the subcanopy of their parental sporophytes. Differences in light response is also observed within species occupying different habitat, like this red alga, Soliaria pacifica. So in general, this red alga can be found in the upper subtidal zone around 3 to 5 meters deep. However, this alga has also been collected from the seafloor at the depth of 35 meters. So we tried to compare the photosynthetic irradiance responses of these two samples. And of course, the light environment at 35 meters deep is different from that in shallow waters. Thus, the distinct photosynthetic performances and light adaptations of the two ecotypes. So in summary, temperature response on the photosynthesis of seaweeds is characterized by a single peak at optimum temperature. The temperature optima for photosynthesis vary among species, which corresponds to the ambient temperatures at their respective distributions. So we have the subarctic kelps, which have minimum temperature for or optimum temperature for photosynthesis at around 20 degrees Celsius, while the tropical Capophycus have it at 30 to 32 degrees Celsius. They are also likely close to threshold levels for thermal inhibition given the decline in their photosynthetic activity above such optimum temperatures. And temperature optima of species that have a heteromorphic life history may not always vary, as in the case of kelps and the cladocyphone, suggesting their overlapping occurrences. Finally, for irradiance response, high light adapted species need strong light to saturate photosynthesis, while low light adapted species can saturate under relatively low irradiance levels and so occurs in deep waters or shaded habitat. Seaweeds may have both light response strategies in one species, as in the ecotypes or life history stages. For example, we have the red algae possessing accessory pigments like phycoerythrin, which actively contributes for efficient light capture for photosynthesis and so are able to grow and live at greater depths. And Maristotheca papulosa, as an example, that reveals its optimum photosynthesis under green light as their phycoerythrin pigment content increases, and so it is able to absorb green light for photosynthesis. So to end my presentation, I would like to thank the following people and institutions for their valuable contributions and support to our research, including those during my PhD in Japan. And lastly, to my current work unit, the University of the Philippines, Visayas. So I've just returned from Japan last year and going back to the Philippines, our beloved country. I have high hopes of contributing something beneficial for the seaweed industry in the Philippines. I think that ends my presentation.
All right.
Thank you for your attention.
All right. Thank you. Thank you very much, Doc I, for the wonderful presentation. We will entertain questions later after our second speaker. So, Doc I, just stand by for the question and answer. All right. So we proceed now to our next speaker. So our next speaker is currently a technical assistant in the analytical services of the School of Agriculture and Food Sciences at the University of Queensland, Australia, where he also took his PhD focusing on natural products and culture of microalgae. A registered chemist where he took his bachelor degrees in chemistry and applied computer systems both from Ateneo de Manila University and his Master of Science in Chemistry on the same university. Also a former research and teaching assistant in the Department of Chemistry from the same university. A very prolific scientist, as well with a total of 13 research articles and counting published in ISI and Scopus Index Journal, focusing on microalgae culture, cultivation, and natural products. Today, he will talk about the development of multi-product microalgae bio-refineries. I am pleased to introduce to you Dr. Elvis T. Chua. Sir Elvis.
Morning. Thank you, Doc JB, for that wonderful introduction. Let me share my screen. Okay, so I hope everyone can see the PowerPoint slide properly. So thank you everyone for joining this webinar. So I trust that everyone is well, especially in this pandemic that we're experiencing right now. Okay, so the title of my talk is Development of Microalgae Multi-Product Biorefineries. So we know that there's already heaps of complex problems, and that is really because of the increasing world population. So according to the United Nations, this is the data from 2015. So in 2090, so it is expected that the world population will reach about 11 billion people. And so this increasing world population has caused different problems, which include our need for energy. And so we have air pollution because we need to burn more coal to get more energy for electricity. We have increasing traffic, and we have deforestation because we need more space to grow our crops. So because we don't have enough space, then there's hunger in the world. And finally, we also have overfishing. So microalgae have been thoroughly studied as an alternative biofuel source. So this was in the year 1990s. And this is mainly because of their high oil content. So here in this table, you can see that with microalgae, it contains like a magnitudes higher oil yield compared to the terrestrial crops. So it's really one of the advantages of using microalgae for biofuel. Another thing is because they are microorganisms, they have high growth rates, and they don't need arable land because they grow in water. And this water can be either fresh water, brackish water, seawater, or even wastewater. In addition, microalgae also contain other high-value products. So with the decrease of world oil price, microalgae researchers have shifted their focus from growing microalgae for biofuel into growing microalgae for high-value products. So for example, here are a few species like Hematococcus pluvialis, which contains high amounts of astaxanthin, and it can reach as high as 4% of its biomass. So astaxanthin is considered the most powerful antioxidant that available in the world. Okay, so another thing would be Pablo valutheri, which has DHA, so which is really important for brain development. So for my case, I worked with Nanochloropsis, which has icopentaenoic acid or EPA, and this is also another omega-3 fatty acid. So Nanochloropsis is a unicellular microalgae, and it's in part of the class Eustigmatophysae, and it is found in both marine and freshwater sources. So it is 2 to 5 micrometer in diameter. So Nanochloropsis is very attractive to researchers because it has very high amounts of lipids and also proteins. And from these lipids, it also has a very high amount of EPA, omega-3, which can be as high as 40% of its total fatty acids. So here I'm showing you some photos from the University of Queensland Algae Energy Farm. So this is where we are able to grow Nanochloropsis at large scale. So however, with past experiences of growing microalgae, especially Nanochloropsis in large scale, we noticed that during the summer season, temperatures in Brisbane can reach as high as 33 to 35 degrees centigrade. And this causes the amount of EPA in the cell to decrease. Hence, part of my research was to find a solution to this. So for the first part of my talk, it will be about how can we how can we solve that problem of decreasing EPA. So usually for culturing microalgae, researchers would look at changing the culture media composition, looking at the temperature and the pH, or even doing stresses like nutrient starvation. So cold stress has already been shown that it can negatively affect the membrane fluidity. So which means at low temperature, we have high membrane rigidification. And so cells counteract this effect by increasing the amount of polyunsaturated fatty acid. Also, with light intensity, researchers have shown that with high light, we see a decrease in the amount of EPA or omega-3. And this is mainly because of the size of the chloroplast. So because EPA is an unsaturated fatty acid, it is commonly used by the cell for the cell membrane or the chloroplast membrane. So thus, we tested this hypothesis and see if the same thing happens with nanochloropsis. So here I'm showing you the method on how we were able to test it. So we initially cultured nanochloropsis until the early stationary phase, after which we transferred it into fresh medium. And then for the control group, they were kept at hot temperature, so which is about 30 to 32 degrees, while those, the treated samples, one group was placed in dark and another group was placed in low temperature, and this was kept for 3 days. So upon 3-day cold or dark incubation, we have seen that there was an induction of EPA. So as you can see here, so 30C means it was the control group, so kept at 30 degrees. And upon, after 3 days at 15 degrees, we have an increase of this omega-3 fatty acid. So this is EPA. And the same thing, even if the algae is nutrient starved, so this is nitrogen starved, so we can still see the same increase. So same thing if we add additional carbon and if it's nutrient starved, but with additional carbon. So the same trend was also observed when we kept the algae in the dark for 3 days. So there was also an increase in the EPA. So because our lab has also the capacity of looking at carotenoids, we then submitted samples for carotenoid analysis. So carotenoids are also antioxidants, and this will, like having high amounts of carotenoids in your final product will also add value to it. So the same trend was observed. At 30 degrees, we have this amount, and then at 15 degrees for 3 days, we have an increased carotenoid. So the same thing also for the dark treatment. So then we had a look at the cell. So what is happening inside the cell? So changes in the chloroplast were observed for the stressed cells. So stress means that they were treated. So it means, stress cells means that they are kept in a high temperature. So as you can see here for A, so this is the control group. So see how the chloroplast is smaller than those that were placed in the cold and in the dark. So this increase in chloroplast means that the cell has to produce more galactolipids or phospholipids, and a high amount of EPA is found in this galactolipid. So, but it's not enough to just, be able to find a way to culture nanochloropsis. So unlike macroalgae or seaweeds where you can just pick them by hand, microalgae need a specific method for you to be able to harvest them. So the common ways to harvest microalgae are filtration, centrifugation, and flocculation. So for filtration, there is the problem of the filter getting clogged easily, and you end up spending more energy and more labor to be able to wash the filter and then put in the next batch. So similar thing for centrifugation, because we don't imagine harvesting like 200,000 liters of microalgae culture, then you'll end up spending so much energy to be able to centrifuge everything into an algae biomass. So for my case, I investigated flocculation. So flocculation is still considered as the low cost, low energy way of harvesting microalgae. So in flocculation, we have to use flocculants. So how do flocculants induce the production of microalgae flocks? So imagine if these are microalgae. So it's already known that microalgae are negatively charged. So flocculants are usually containing positive charges. So when they are mixed in solution, the negative charge around the microalgae are neutralized by the positive charge of the flocculants. And so this induces the algae to come closer so they can form huge clumps. So this is via charge neutralization. Another way is if we use some compounds which also have positive charges, so maybe tiny polymers. And the same thing. So this is this mechanism is called as electrostatic patch. The third one is if we have a very long polymer. So with this, the polymer somehow acts as a bridge. So this is called interparticle bridging. So for my case, what the flocculation method I used was we culture the microalgae in bags and then transfer them into jars and then add in the flocculants and then mix them and then allow them to settle for 30 minutes. And then we just measure the absorbance, which tells you how much microalgae is left in the solution. So the common flocculants that are used are aluminum sulfate and iron chloride. So these are inorganic flocculants. And as you can see in this graph, we need about 87.5 ppm to be able to obtain greater than 95% flocculation efficiency. So this is okay if the plan of your final biomass is maybe for biodiesel. But if we want to use the final biomass for human consumption, then the high amount of aluminum or iron may not be good for our health. So another type of flocculant is what we call bioflocculants. So previous researchers have already tested the use of chitosan, but results were a bit were a bit different between researchers. So we investigated this. So first we tried, and as you can see in this graph, even increasing it to 25 ppm, we only got 20% flocculation efficiency. And if we go back to the chemical structure of chitosan, we see that it has amine groups. So which means you need to find a way to activate the amine groups because we need a positive charge. So we conducted the central composite design experiment to optimize 5 different variables. So here they are. And we found the optimum value here. And as you can see, there is a need to decrease the pH of the solution. So that is so that we are able to protonate or induce the positive charge into the chitosan. So we tested it at larger scale. So at the top here is 10-liter culture, and this one is a 5-liter culture. So another thing is chitosan can also be extracted from mushrooms. So as there's numerous papers that have already proved that you can extract chitosan even from the mushroom waste. So we also tested this and prepared chitosan from our lab and tested it. First, we characterized the chitosan we got via IR spectral analysis. And as you can see in this graph here to your right, so the top part is the chitosan that's commercially available, and this one is the mushroom chitosan. So all the peaks that are present in the commercially available chitosan is also present in our mushroom chitosan. So then we tested for its flocculation efficiency, and as you can see in this graph, so the chitosan that we purchased from Sigma, so which is a special company, gave similar or the same, actually almost the same results as our mushroom chitosan. So we tested this mushroom chitosan in the farm, so in the pond. However, our results have shown that there's only 73% flocculation efficiency. And this could be because of the mixing of the chitosan into the culture. Because when we tested it into our 2-liter tower, we can see that there is high flocculation efficiency. So with proper mixing, mushroom chitosan can be used to harvest microalgae. So the third part of my talk is about how do we extract the oil from the algae. So this talk is about producing multiple products or obtaining multiple products from nanochloropsis. So the first one would be the omega-3 rich oil. So traditional extraction methods use chloroform. And as we all know, chloroform is a very toxic chemical. So numerous methods have also been investigated, and I categorize them into 4. So first is using solvents. Second is using a non-chemical method, which may include osmotic shock, thermal, or enzymatic. The third one is using supercritical CO2, and finally ionic liquids. So in our lab, we have tested and showed that osmotic shock, thermal, or enzymatic methods are not possible with nanochloropsis, and this is mainly because of its very thick cell membrane. So here you can see that ethanol can actually be used to obtain the same amount of EPA and oil from nanochloropsis, and we compared this with the common method, which is chloroform method. So for supercritical CO2, here you can see that it is very low as compared to the previous example. So, and this was already done at 300 bar, 40 degrees in 2 hours. So supercritical CO2 extraction is really a very highly energy intensive method. So it may not be suitable for nanochloropsis, but maybe for other algae, it might be okay. So the third method that I tested was ionic liquids. So during the time that I was doing this, ionic liquids was a big thing. So what are ionic liquids? So they are actually ionic compounds. However, they are liquid at less than 100 degrees centigrade. So because of this, they are very attractive for extraction. So ionic liquids have also been tested to extract various compounds from microalgae. So here we have a paper where they extracted azacanthine from Haematococcus. And as you can see, it is actually better than using the traditional organic laboratory solvents like acetone and ethyl acetate. So the same thing was also tested in Chlorella vulgaris. So I prepared the ionic liquids in our lab. And this is just by mixing collinium hydroxide and amino acid. So my results show that choline arginate is an efficient solvent for lipid extraction of nanochloropsis. So this is compared to all the other amino acids. And it was still even effective, even at room temperature. So what is it with choline arginate? So first is it can form the most number of hydrogen bonds. And the second one is it forms the strongest hydrogen bonds. So why is it important to have this property? First, it is because choline arginate is able to break the hydrogen bonding network of the cellulose based cell wall of nanochlorosis. So here you can see the cellulose network that is being proposed in a cell membrane. So in the presence of choline arginate, it is able to break or disrupt this network such that the cell breaks and then you're able to extract the lipids out. So for my last last part, it is about extracting the proteins from the protein-rich defatted biomass. So a common method is to extract proteins via alkaline treatment. So we tested this and optimized using 4 variables. So here we can see with the optimized parameters, we are able to extract 80% of the proteins. However, recovering is also a different problem. So proteins can be recovered by isoelectric precipitation, and this is already a common method, especially in biochemistry. So here you can see at different pH, the color of the supernatant changes. So from pH 5 to pH 4, there's a big change in the color. So, however, when we tested the precipitate, it only contained 30% protein. It was 30% protein. So it means 70% was something else. So this means that more research needs to be done in this field. Another method that can be used for extracting both lipids and proteins is 3-phase partitioning. So I'm going to play. You can see here how quickly the phases form. So this is in real time. And the hope for this method is really to have the proteins precipitate in the middle, and you have the lipids that are extracted by the alcohol. So I tested different alcohols, ethanol, isopropanol, and tert-butanol. And here you can see the top layers, the alcohol layers to your left, and the remaining layers to your right. So as you can see in the interface, the biomass is still very green. So which means more optimization will be needed for this method to be made sustainable. Now, aqueous 2-phase systems, so which is what 3-phase partitioning really is, may not work in some cases, but it works well in other cases. So here we just tested extracting phycocyanine from spirulina. So as you can see, by changing the volume of the different components, we are able to concentrate the phycocyanine into a very tiny volume. So for future research, it is important to look at the cell holistically. So by holistically, I mean preparing or designing a multi-omics approach. So this involves doing lipidomics and metabolomics and maybe transcriptomics. That way, we are able to see what is really happening inside the cell and maybe find a way to control a specific pathway. Then we can further optimize the production of our intended natural product. So another field to look at would be multi-step fractionation. So if we are able to fractionate the different products that we want from the microalgae and have a higher purity, then that would be very ideal, especially for industry. So here, this is from a paper where they propose a method to be able to extract and separate pigments from proteins and from sugars. So in summary, microalgae biorefinery should really include the culture and induction of the microalgae, the harvesting method, and the extraction of the products. And the choice of the methods, including the microalgae species, really depends on the final product of interest. So here I'm just sharing with you two different biorefinery processes that are already published. So here to your left is a biorefinery process for the extraction of fucoxanthin, which is a carotenoid, EPA, and chrysolaminarin, which is a carbohydrate. And this is from the diatom thiodactylum, tricornutum. And to your right is another biorefinery where they are able to purify the PUFA-rich oil. So PUFA is polyunsaturated fatty acid, and able to separate the saturated fatty acids and the monounsaturated fatty acids from the polyunsaturated fatty acid. So I would like to thank my advisory team. So Professor Pierre Sheng, Dr. Sky Thomas Hall, Dr. Christiana Dalmolin, Dr. Michael Netzel, and Dr. Gabby Netzel of the University of Queensland, and two visiting academics for their assistance and very helpful brainstorming sessions. So Dr. Elada El-Tanahi from Mansoura University in Egypt, and Dr. Ajam Sheikh from India. And I also would like to thank other staff members of the University of Queensland. So Dr. Ann, Dr. Hung, Dr. Nana, Dr. James, and Dr. Evans. And finally, Professor Rob Adkin and his team from the University of Western Australia for their help with the ionic liquid experiment. So thank you very much. And I hope you have a very good Friday ahead.
All right, thank you very much, Doc Elvis. So may I ask the two speakers to be here together so I could able to ask some of the question. But before we do that, we would like to inform everyone that we are well represented from different islands in the Philippines, from Luzon, Visayas, Mindanao, and also we have also from overseas. All right, so before the question and answer, I would like to just inform everyone that we are being viewed in Bacolod, Visayas State University, Surigao del Sur, Dumaguete City, before Giwan, Mindoro State University. We have in UPV Miagao, Valencia City in Bukidnon, Zamboanga del Sur, Panabo, Davao del Norte, Sorsogon City, Pangasinan State University, General Santos City. We have one from Korea, South Korea. We have Misamis University and Ozamis City and San Jose Occidental, Mindoro. And we have also from Leyte. All right, so are you guys ready for the question and answer? I think we could just be relaxed or whatever. All right. So first is it's just only a basic. I think both of you could answer this. So, what is the basic principle of classification of algae in various classes? So, anyone could answer?
May I
Doc,
answer?
go ahead, Doc.
Okay. Well, pretty good question, although it's not that related to our topics, but how do you say? Somehow basic for anyone to know. Well, we usually classify algae in terms or in terms of their in terms of the presence of the chlorophyll pigment or the pigment that they possess. So algae may be classified as chlorophyll or Phaeophyceae, Rhodophyceae by the by the presence of pigments. Like for chlorophyceae or the green algae, they contain chlorophyll A and B. The brown algae or the phaeophyceae contain chlorophyll A, C, and xanthophyll. While for rhodophyceae, they possess phycobiliproteins or like phycoerythrin in the aside from chlorophyll. I
All
hope that
right.
answers the question.
Sir Elvis, do you have any additional to that?
Yeah, I think Doc Iris would be the best person to answer this because she's really into the basic science research, but mine is more applied. But anyway, I think yeah that is correct. And this is also what I have seen from the different papers I've read, especially with microalgae. Like we can have atoms and chlorophyta, yeah, these things. So yeah.
I think very debatable ang classification ng algae, but I think yung common way of doing it is classifying it into pigment because it's more straightforward. But I know previous there's a very controversial debates on how to classify it. But I think for now, it is more straightforward that we will classify them according to pigment content. So I agree to the both of you. All right. So let's just have some series of questions here from that I've listed. So can I give the floor first for Irish? So this some of the questions that I've got. So with the ecophysiological profiles of these seaweeds, what can we do with this in terms of cultivation this for mass proliferation?
Okay, that's a great question. Using these profiles, we could, getting those estimates of optimum conditions, it is great to have these profiles so as to suggest or recommend also conditions, optimum conditions for their growth, for maximum or for mass propagation. So, although, as I mentioned earlier in the report, we get results that are higher than the ambient temperature or uh, higher than their optimum range for growth. I guess the optimum temperatures that we get from photosynthesis could still be used. We consider them as the maximum temperature. So maybe values exceeding those optimum temperatures would be critical already for their growth. So
Mm-hmm.
getting those data or information, it's very helpful on the culture side. Mhm.
Doc I, so if the photosynthetic response is above their optimum, is it possible that this could be Q as possible adaptation of this specific seaweed species for climate change?
Yes, definitely. With well, currently we're experiencing climate change, and the organisms, we also do adapt to changes. So there are possibilities that in the future this optimum temperatures could change as well. But this time, the values that we get or we obtain from the experiments could still provide information as the maximum temperature, or let's say
All right.
irradiance.
Okay. So thank you for that. So I have a question for um, Doc Elvis. So um, what could be the implication of um, the light intensity on the size of the chloroplast in nanochloropsis? So what could be this? I mean, what is the implication for this if you're in terms of you're going to culture it or if you're
Oh,
going to take extract?
okay. So really, from my seems that the algae is able to adjust to the stress after 3 days. So it means, for example, like here in Brisbane, because during the summer, as I said, it's very hot and then light intensity is very high. So it could be that before we even harvest the algae, or if even before we do extraction, we can keep it in the dark for 3 days, and then after that's when we harvest. Then we have a better quality biomass as compared to just harvesting it immediately and doing the extraction. So
go ahead.
Sorry again. Yeah. So the same thing with the temperature also.
All right. So Doc Elvis, just a follow-up question. So if the light intensity has an effect on the size of the chloroplast, so does it mean that photosynthetically they are also affected?
Yeah, so actually, this is something that also my supervisor was asking me. So does that mean that they're not growing? Because it will be in the dark for 3 days, right? But then if you can, if you remember from my method, the algae is already in its early stationary phase. So we isn't any growth happening anymore.
All right.
So
okay. So this question could be answered by both. So could GMO algae improve the production for the use of biorefineries? Is there any application here in the Philippines? So could you answer first, Doc Elvis, for this one?
Yep. So this question is a bit, even here in Australia, it's a bit controversial, I guess, especially when you talk about GMO here in Australia, because of the very harsh, or I would say harsh or strict biosecurity. So if you start growing GMO algae, like there's so many documents that you have to fill out such that people don't into that. So for my supervisor, this is his goal. Can we try and produce like good quality algae without going into editing the genes?
Yeah. So you are advocating like a natural way of cultivating it, like of what you're doing also, already in Australia.
Yeah. But there's also another group here that's looking into GMO. But then if it's GMO, the production, the way you produce it at a large scale will be very expensive, because you have to maintain proper biosecurity protocols.
All right. So follow-up question with you, Doc Elvis. So if you're, for example, if you're going back in the Philippines and you're going to use the technology that you've learned in Australia, what could be like a potential microalgae that could be used for biofuel or other byproducts from microalgae?
Yep. So I think this really depends on where you want the pond to be, the large scale pond, because what we found here is it's best to isolate the algae that's local in surrounding areas, so that they're already used to the temperature, to the to the environmental conditions. Because otherwise, because we had experiences here where we like ordered some algae species from, say, Tasmania, where the temperature doesn't even reach 30 degrees, or very rarely. So it's going to be very hard to grow it in Queensland. So I would say it really depends on where you want to grow, or to have the pond.
But just a hypothetical answer. If I mean, in the Philippines, if you're going to do that, if you're going to do that here in the Philippines, what possible microalgae that you like to work with?
So if it was me, I might look into, say, if it's in Manila, maybe look at try, if it's for biofuel, maybe
Mm-hmm.
try using wastewater. So with
Mm-hmm.
wastewater, usually you want freshwater algae, so like chlorella or
Mm-hmm.
some cyanobacteria like spirulina. But then it's spirulina, you don't really want to do biofuels for spirulina because lipid
Mm-hmm.
and is really low.
Yeah, yeah.
But maybe if you're close to the sea, say coastal region, so
Mm-hmm.
look at marine algae. And
Mm-hmm.
mm, if you're able to purify, as what I said in the last part of my talk, if you're able to purify the polyunsaturated from the saturated fatty acids properly, so
Mm-hmm.
it's okay to look at nanochlorosis.
All right. So we are looking forward if you're going back here in the Philippines soon that you'll be able to
Yes.
establish this technology. So I mean, I think it's very helpful because one way of culturing it and also solving environmental problems specifically in Manila Bay. So I think we are all looking forward for that. All right. So next question is for Doc Irie. So this one is from Jose Avila from Chuseon University in South Korea. So regarding studies on gametophytes of kelps, have you found different responses on the male and female gametophytes?
Um, well, in our experiment, we usually do not separate the male from the female gametophytes. In fact,
Mm-hmm.
we had some problems uh, in obtaining the right amount or appropriate amount of replicates of gametophytes. So we haven't tried comparing the photosynthetic responses of the two, but perhaps there might not be any difference because in nature both the gametophytes are together in
Mm-hmm.
the we don't separate them. And just one thing also to note, well, although we get results for the results on the gametophytes, it does not necessarily uh, speak on the reproductive success of the gametophytes. It's a completely different study that we still need to look further because there are cases that, well, they are alive, they do photosynthesize, but perhaps when they do fertilization, they can no
That will be
longer,
a different thing.
yes, they can no longer grow, or maybe there's no offspring after despite the fertilization.
Mm-hmm. All right. So basically, you're saying that um, hypothetically there will be no differences on their photosynthetic response, correct?
Yes.
All right. So another question for you, Doc I. So what could be the implication of ocean acidification in the light capture efficiency of red and brown algae?
Pardon? Ocean?
Um, Ocean acidification on light capture efficiency of red and brown algae.
Okay. When we speak of ocean acidification, I think it's more on the change in pH. So perhaps the seawater would get a lower pH. And there are studies that algae in general are better in with low pH. So I guess the greater the amount of algae we have in the coast, it would definitely help in mitigating effects of ocean acidification.
Well, actually, yeah. I mean, in ocean acidification, marine plants are the winner during this phenomenon, actually. All right, so another follow-up question, Doc I. You mentioned that some brown seaweeds have different life stages that co-occur throughout their known growing season. So what are the advantages or disadvantages of the strategy for this type of taxa? Is it safe to say that in this case, the microscopic stages are low light adapted? Is this the case with other taxa with alternation of heteromorphic generation?
All right. With regards to Cladocyphon or Mizakii and even kelp species, they have the same life history stage wherein they have the same heteromorphic life history stage wherein they have the macroscopic sporophyte and microscopic gametophyte. So being microscopic, most once the macroscope or reproduce mature sporophyte sporulate and release these gametophytes, these spores settle in the substrate. So definitely we could you think of it as the gametophytes being low light adapted species. So being also microscopic and their cellular structure may somehow be more sensitive than that of the macroscopic sporophyte. So it is advantageous for them to be low light adapted because with high irradiance levels, definitely these microscopic gametophytes um, would get affected and result to photo damage. So they
Mhm.
will no longer grow.
And
Unlike
I
with
think they will.
macroscopic sporophyte, they do get exposed at higher radiances and maybe they could tolerate higher radiances.
Mhm. All right. Okay. So thanks, Doc I. So I'll just move with Dr. Elvis with this question. So um, I think this is directed to Dr. Elvis. So how many biomass of yield 1 kilo or 1 liter of oil or whatever product it is desired for? So I think is it species specific yung yield of the biomass?
For the oil, for the biomass, of course. So yes, so this is species specific. So
Mhm.
because microalgae have different sizes and the weight is also different. So yeah.
All right. So I have a follow-up question with Doc Chua. So what could be the potential economic values of these extracted substances from this algae?
So so here in Australia, really, there's a few algae companies that are, startup companies that are already, um, how do you say, like being set up. Okay,
Mm-hmm.
and so most of the companies are really into looking at the omega-3. So because of the yung fish oil industry, so this is
Mm-hmm.
the profitable market.
Mm-hmm.
So that's why like even in our group, that's why we want to look into non-peroxis, which are
Doc Chua, wait up. We're hearing background. Sorry. Oh, all right. Okay. So go ahead, Doc
yeah.
Elvis.
So yun po. And then maybe there's actually like a table of different natural products that you can get from microalgae, and most of them are really high value. So if you go
Mm-hmm.
say phycocyanin from spirulina, so
Mm-hmm.
these are fluorescing compounds. So because of their property, fluorescence property, they are highly if you are able to produce like high purity phycocyanin, you can use it for preparing molecular probes or stains for
Mm-hmm. Mm-hmm.
diagnosis or something like that.
Mm-hmm.
So yeah, yun. So it really depends on the purity, number one,
Mm-hmm.
and yung property of the compound itself. So most compounds that people are after are bioactive compounds. So kasama na din yung say astaxanthin. So especially astaxanthin.
Yeah, kasi in the Philippines, medyo siguro list or I mean neglected industry ang microalgae because it's possible na walang market or wala pang technology. But if you're going to make it very attractive in the Philippine setting, ano yung potential na pwede niya maging byproducts if you going to market um, this microalgae in the Philippines?
Actually, there's already a few companies in the Philippines, but usually
Mm-hmm.
sell the microalgae as a pure biomass.
Mm-hmm.
I remember before when I was still in the Philippines, there was a company selling coffee, 3-in-1, pero may spirulina.
Mm.
It's like they mix it together, so as a biomass. But yeah, I don't know, maybe It really depends. Pwede tayong mag-biofuel or I know in UP Biotech, UPLB Biotech, I know that they are thinking about preparing microalgae for, say, fertilizer, but that's also of low value.
Mhm.
Usually, kasi, if you try and prepare and sell microalgae as a pure biomass, it is considered like a low value product.
So dapat yung extract or compound from the microalgae.
So really, kailangan yung extract if you can sell it at mas ano siya, mas high value.
Okay.
If you imagine, if you can prepare fish oil capsules, but then it's algae coming from algae, so you can actually find it already in the US, and I sell it at like one capsule for $30. So which is really very expensive.
So possible ba, Doc Elvis, na we could produce it here in the Philippines, that the market is the United States or other countries?
Yes, possible.
Kasi we already doing this in seaweed industry, so we do the raw materials and we send it out for abroad companies. So so when you're coming back in the Philippines, you're going to help the microalgae industry for possible production for this in the market.
Hopefully.
So excited na kaming bumalik ka, no?
There is a group.
All right, so I'll go back now to Iris. So there's a couple of three questions here for you. So what is the practical application of knowing seaweed ecophysiological profile in optimizing the production of important natural product derived from seaweeds?
Sorry. Ayan. Again, sorry.
Okay, so
Nawala kayo. Sorry.
it's okay. So I think the question will be, if we already know what is the implication if we already know the seaweed ecophysiological profile in if you're going to optimize production of derivative products from seaweeds.
Okay. One thing is that with this profiles or information on their photosynthesis, it would be advantageous para sa production because definitely photosynthesis is directly related to growth, and from growth we get biomass. That is one, but it's also different thing for in getting or deriving products from it. But definitely, pag maraming biomass, marami ka rin makukuhang product out of it. So that is one. But the effects would also be different. Again, you could manipulate your organism depende sa anong target ninyo. So for example, as I mentioned earlier, exposing at different light wavelengths. So for example, in Japan, they wanted colored red. So perhaps after harvesting their samples and they get, let's say, light colored Meristotheca populosa, they just have to, I guess, one suggestion that I could give based from the results that we have, they could expose the seaweeds to, let's say, 3 to 5 days in green light, then they get the red colored product already. So somehow you could get from those results, you could get some inputs in suggesting innovations also. Depende sa kung ano ang target na product.
Yung objective mo, no?
Yes.
So you mean that when this red algae increases the color, so meaning it increases the pigment content of it, right?
Yes, definitely. But another case, I think I've seen one question here from the participants regarding Eucuma and Capophycus, and the product that they release is, well, we get carrageenan as product from these seaweeds. So well, it's different in the case of growth kasi we do plant or cultivate Capophycus for 45 days. I guess the first 45, not 45, but I guess the first 30 days or 21 days, it's for growth. Palakihin muna ang seaweed. But when it comes to the carrageenan that we get, they have to be matured enough. So that's why we extend it to 45 days. So perhaps let's say sa growth rate then ng isang any microorganism, nasa growth phase yung lag phase. Tapos pag nakaabot na siya ng stationary phase at the end, doon na nagmamature ang carrageenan, which is our target.
Okay, so thank you, Doc I. So I'll go back to Elvis for this quick question. So if we are to use freshwater microalgae strain for mass cultivation, can you give us some practical tips that we need to consider in establishing a large-scale setup here in the Philippines?
Number one, of course, would be if you have a freshwater source close by. Say you don't want to use wastewater. Kasi you don't want to use freshwater, especially kapag the freshwater source is being used for people, right? Because we don't want to have like an algae versus food problem again, which is what was happening with the biofuel from terrestrial crops. Another thing is, of course, you need like small scale lab experiments before going to the big scale ones. So if you can still remember dun sa slide ko, in our UQ algae energy farm, we have those big towers, 200-liter towers. So before we go into that 200-liter towers, we would start in a small 20 ml flask. So it's a 20 ml flask, and then we go into big bags, 20-liter bags. And then after that, we go into the tower. And then yung tower, yun yung papunta sa pond. So fortunately, for microalgae, we don't need 30 days to grow it.
Yeah. It's much faster in microalgae. Yes.
So
you optimize first on small scale, then you multi-scale it up, correct?
Yes. Of course, the way you optimize at the small scale, you have to take into consideration the environmental conditions where you want the algae to be, the large scale farm to be at. Kasi maganda yung growth at 15 degrees, but then umabot ng 15 degrees unless maybe you have your pond in Baguio or which then you'll have a problem of like how would you bring the water up to Baguio, or may enough space but to have this big ponds. So these are the considerations, I guess.
So with your experience, roughly how much it is to establish a macroalgae farm?
By much, you mean money?
Yes, how much? I mean, if there's an interested like,
I don't know really. No idea.
All right.
Kasi it will also depend on what product you want, right?
Yes, exactly.
Even dito sa energy farm namin, we still haven't got into large scale extractors. Expensive equipments, and you don't want to buy just buy anything and then test it there. You want to make sure that whatever you buy is really
So I will just to follow up that question with this question. So is there any chance that microalgae based biofuel can be feasible since the cultivation process is expensive?
Okay, so that's why our group proposes a multi-product bio-refinery. So you just grow microalgae just for biofuel. Say if to produce biofuel, that's okay. But make sure that you have another product that is of higher value to extract. So make biofuel as like a second priority. And higher value as well.
So I hope from our participants here, some of them probably are students, researchers. So I think this could encourage more to study microalgae and of course seaweeds, because basically we are doing mass production of this for their extracts and substance that we are getting from.
Yes.
All right, so I think we are concluding this webinar. But before I end this Q&A, I will give this question to both of you. This is some sort of Miss Universe question. So how will you answer this? So the first question will be, will algae interferes with human health and environmental problem if it's produced in large scale?
May I answer first?
Go ahead, Doc I.
What a Miss Universe question.
I know. There will be a follow-up. So just stay put.
Well, I disagree. Doesn't affect, I mean, we don't get any disadvantage in growing seaweeds at large scale, whether microalgae or macroalgae. Because this time we have the earth is too, how do you say, there are a lot of people now. We have a big population and we need food and in order to survive. And I think meat to get protein source is not that enough. And with algae or seaweeds, which is also a rich source of protein, it's definitely helpful. I mean, it's a good alternative.
All right. So, Doc Elvis?
For me, I think it can be, especially if the people who are going to set up the large-scale farm didn't, like they just thought about it one day and then just went out and then maybe started digging for a pond. If you don't do proper research or proper literature search, yung mga ganun, then it can be harmful.
All right.
Actually, because like in the Philippines, we already have problems with space. So I really think that the only way to have this sustainable production is when people come together, collaborate. So we need, not only phycologists, but we will need like chemists, engineers, and even economists, to look into yung cost. So usually sa microalgae research, this is something that people don't think about kasi we are only after the production and we don't really care like magkano ba yun? Like is it worth spending so much time and money into research and then in the end hindi mo rin naman pala mabebenta.
So basically it's more of like looking into multi stakeholder cause and effect of the process, right? Hello?
Yes, that's correct.
All right. Okay. So the last question is actually nasagot na ni Doc Elvis, but I'm going to give it to Iris. So how to sustainably produce large-scale seaweeds or algae?
Okay. Sustainable. Okay. Sustainable aquaculture. Definitely there are, it's as mentioned earlier ni Doc Elvis, lahat science-based. So we do research, we get information from those research para ma-apply siya sa industry. So in the case in the Philippines, we do have seaweed production mainly on carrageenophytes. So how to achieve sustainable aquaculture of these carrageenophytes? One is we definitely have policies, no, and these policies are based on research as well. So one is, I think, is aside from, number one is research. Second is strict implementation of these policies or strict implementation of recommendations coming from these researchers. Kasi in the long run, if we do not follow these recommendations from the results that we get from research, everything will crash as well.
Yes.
Mm-hmm.
Of course, with the if we're doing research, in a way, this data or this input could be we could have a predictive capacity if ever this will happen in the future. At least we already know that we have preliminary information about this, right?
Yes, and definitely from those we could get. I mean, we could respond immediately.
All right. So, Doc Elvis, do you have addition to that?
Um, I think we also need innovative researchers these days
Mm-hmm.
because before I finished here, I was actually thinking about how is it that we're able to produce this big farms when I go back to the Philippines, especially because space is already a problem. But
Mm-hmm.
there are researchers like I don't know if you know Professor Coelho. So he is in he's actually a bioengineer from the US, and they are able to produce microalgae at a large scale without the need of like having big ponds, so big air.
Mm-hmm.
So they just grow the algae in bags, and they have a specific like a special what do you say like a special engineering of growing the algae in bags, and
Mm-hmm.
then um, like the culture can flow properly. So these are the things na kailangan I think kailangan ng algae research.
So, basic research plus innovation, no?
Yes. So kailangan there should be a merge between basic and applied research.
Sana mas marami tayo ma-encourage from our participants na we could do more research on algae. Actually, honestly, speaking on personal note, so parang sa Philippines yung phycology is medyo parang hindi na siya napapansin. So most of the other researchers will go to corals or fish, but if you're going to look on it, phycology, there's a lot of benefits from it as food and byproducts or extracts or compound that you're able to get from it. So I think we need to encourage more students, researchers, to do studies on seaweeds and microalgae. So I think I hope agree naman kayong dalawa doon, no?
Yes, I agree, sir. Especially kasi di ba, we're an archipelago. So
Exactly.
we will have like whatever algae is being researched in other countries, most likely we will have it. Kasi yung ocean naman, it's not static. So
Exactly.
they're very small and they can spread around. Yeah.
And syempre, some of our former supervisors are retiring. So we really need a lot of human resources to do studies on phycology. No? So Doc I, do you have inputs on it?
Well, wala na po. Ano lang, tulungan lang po tayong lahat.
Exactly. More collaborative work because we cannot do it alone. So, all right. So, with that, I would like to thank Doc Iris and Doc Elvis for gracing us today for this webinar. But before we end this event, I would like to call in um, Dr. Armin Coronado um, for his closing remarks. So actually, Dr. Armin is a very good friend of mine way back 2 million years ago. So he is now the director of the Institute of Science and Technology Research in Polytechnic University of the Philippines, an associate professor of the Department of Biology, College of Science of the same university, and the current vice president of the Philippine Ecological Society Incorporated. So Doc Armin?
Yes, thank you, Doc JB. So good morning to everyone. So my task this morning is not to have my closing remarks, but to make a synthesis from the two interesting talks that we heard from Doc Iris Berlongan and Dr. Elvis Chua. So first, I would like to congratulate our speakers in presenting important topics in the field of psychology, which consequently makes us understand the biology of economically important algae. I would like to say that we really enjoyed listening to your talk, and we open our minds that there are still many research areas in psychology which needs to be tapped. So Dr. Irish Borlongan discussed the ecophysiology of seaweeds in the Western Pacific Ocean. In the talk of Dr. Irish, she emphasized the two important factors that affects the productivity of seaweeds, the light and irradiance. Particularly, Doc Irish measured the photosynthetic response of seaweeds as adaptive strategy to temperature and irradiance. So Doc Irish made us understand that the maximum photosynthesis of seaweeds in response to temperature would vary depending on their distribution. For example, tropical species have higher optimum temperature as compared with subarctic species. Thus, this optimum temperature is regarded as the threshold level for thermal stress. Moreover, Doc Irish stressed that in the species with heteromorphic life, the occurrence of the two life stages share similar optimum temperatures. On the other hand, Doc Irish also emphasized that the light spans of seaweeds is dependent on how species were adapted to light. For example, highlight adapted species has higher irradiance level, to saturate photosynthesis as compared to low light adapted species. So this was exemplified in the differences on light adaptations of the two ecotypes of red alga Soliera pacifica, which shallow and deep water populations had high and low net photosynthetic rates respectively. So Doc Iris also mentioned that the lighter spans of seaweeds found in subtidal water is dependent on the photosynthetic pigment that may contribute for efficient capturing of light. For example, the picoerythrine in Meristoteca papulosa can be able to absorb green light that makes them adapted to deep waters. On the other hand, there are some species with heteromorphic life history would show different irradiance response. So the net photosynthesis of microscopic stage saturates at lower photosynthetic active radiation or PAR, while the macroscopic stage at higher PAR. So our second speaker, Dr. Elvis Lichua, discussed on the development of microalgae multi-product biorefineries. In the talk of Doc Elvis, he used nanochloropsis as model microalgae to understand the importance of optimizing biorefinery to make microalgae farming sustainable. Doc Elvis pointed out that first, culture condition is vital to maximize the quality and quantity of the metabolites produced by the microalgae. And different stress conditions such as temperature, light, and nutrients have different metabolic responses that would affect the production of the natural product, or specifically the lipid production. Second, harvesting technique is critical in obtaining higher yield of microalgal biomass. So Doc Elvis presented to us the flocculation technique in which in his study, aluminum sulfate and iron chloride can be used as a flocculating agent in harvesting nanochloropsis that would yield around 95% flocculation efficiency, and he also explored the use of a bioflocculating agent, the chitosan, and this could be an efficient also to have a 97% flocculation efficiency. Okay, third, Dr. Chua presented to us the various methods of extract methods to extract lipids that would include ethanolic extraction, supercritical carbon dioxide extraction, and ionic liquid extraction. And These methods, according to Doc Elvis, are efficient in yielding high lipids in nanochloropsis. Fourth, Doc Elvis introduced the three-phase extraction method to obtain protein from the fatted microalgal biomass, in which this method would simultaneously also extract the lipids. However, uh, the low lipid and protein content were obtained in this method, and there would be also some contamination of ammonium sulfate salts. Okay, so the topics presented by the two speakers are equally important, not only in the fields of mycology, but also with other disciplines of applied science. It is clearly important that the physiology of the algae must be fully understood to determine its application. So this finding paves its way that there are a lot of things that we should understand in algae to maximize their full utilization to help improve our quality of life. The bottom line here is that algae have many promising contributions to the society that is usually neglected by the community. So the challenge for us now is how we can sustain valuable research in order for us to cascade what would be the importance of algae in the environment, nutrition and health of Filipinos, as well as the economics of the Philippines. Okay, again, thank you to our dear speakers for sharing their timely and interesting topics on algae. Good day and keep safe, everyone.
Thank you very much, Doc Armin. So now I will be reading the certificates. So the certificate states Biological Research Services Laboratory, Natural Science Research Institute, University of the Philippines, Diliman, and Philippine Phycological Society Incorporated. This certificate of appreciation is given to Iris and Burlongan as resource speaker during the webinar entitled Algae Talk, held on December 4, 2020 via Zoom. Signed, Head of Biological Research and Services Laboratory, Rangeli Vital, um, Wilfredo Wilberto D. Monetilla, President, PPSI, and Jonas P. Kilang, Acting Director of National Natural Science Research Institute. And also, the same content given to Dr. Elvis Chua. Thank you very much, Dr. Elvis and Dr. Iris, for gracing this webinar. So with that, I would like to call in um, for the closing remarks, the laboratory head of the Biological Research and Science and Services Laboratory of Natural Sciences Research Institute, University of the Philippines, Diliman, Dr. Pairang Jelly G. Vital. Doc Pai.
So hi, good morning everyone. I am having a technical difficulty, but anyway, thank you very much, Dr. JV. I hope you hear me now. Right. Good morning, everyone. I am sure that all of you are having a great morning. So please allow me to thank the following. First, of course, our amazing speakers, Dr. Iris Ann Berlongan from UP Visayas and Dr. Elvis Chua, currently in the land down under in University of Queensland, Australia. Thank you for sharing your great works with all of us. So just to share, I met both of them in an international phycology conference, and I hope we could all attend one again soon and form new and lasting collaborations. Second, of course, I would like to thank the Philippine Phycological Society for partnering with us in our lab's fourth webinar this year. So PPSI is headed by Dr. Wilberto Monatilla, and you have heard him earlier, and other board members who equally love algae. If you could also please turn on your video if you can po. Dr. Armin Coronado, you just heard him as our synthesizer or Vice President. Sir Owen Horda, Dr. Fred Santianes, Dr. Tito Evangelista, Sir Eldrin Arguelles, Dr. Elsie Tech, Dr. Emmy Mandia, our amazing moderator, Dr. J.V. Sajo, and Sir Lawrence Vitug. Thank you very much po. Third, this webinar will not be possible without the support of our NSRI Director, Dr. Jonas Kilang, and the efforts of the amazing and hardworking researchers and staff of BRSL. So for this webinar, your co-hosts, as you can see from the participants tab, if you can also turn on your video, please, are Rian, Brian, Jude, Jessa, Donna, Zarina, Ryan May, Erna, Raquel, and Joy Ann. The credit, of course, goes to them. A virtual round of applause, please, for all of our organizers. All right, so the Biological Research and Services Laboratory can be found in the Natural Sciences Research Institute in University of the Philippines, Diliman. We offer different facilities, researches done by different project leaders and different services. So please feel free to email or contact us if you have biological concerns. We have our official Facebook page, which is Biological Research and Services Laboratory. Lastly, I would like to thank all of you, our dear participants coming from all parts of the country and the world, the universe rather, who show great interest and enthusiasm in the field of algae and joined us this morning for the BRSL PPSI joint webinar, Algae Talks. Inviting you also to join us in the Methods of Microalgae book launching, headed by our former PPSI President, Professor Emeritus Milagrosa Gos, on December 9 at 10 a.m. Registration details are in the PPSI and BRSL Facebook pages. So please stay tuned with our BRSL and PPSI Facebook pages for updates. With this, I wish you to have a wonderful and safe holiday. Hope to see you all again in our webinar next year. Thank you very much, and I turn back over the virtual floor to Dr. JV.
Thank you very much, Doc Pai. So now we will have some announcement from your laboratory. So Serena.
Hello, thank you so much, Sir JV. I am Jessa. Again, thank you so much, everyone, for attending today's webinar. Please send us your feedback. The evaluation form is now posted in the chat box. So please evaluate in order to receive your certificate of attendance, which you will be receiving within 1 to 2 weeks. Try to do this soon because the link will close 15 minutes after the end of the webinar. So we will now be having the photo document